Dear students, in this video we will discuss the physical properties of the amino acids. Amino acids are usually colorless crystalline solids having very high melting points. They are usually soluble in water, and in its aqueous solution amino acids show amphoteric behavior. Amphoteric behavior means they react with both acids and bases, and they are always neutral in nature in their aqueous solution. And why they show amphoteric behavior? Due to the formation of zwitter ion. Zwitter ion, as I have told you in the earlier video, that amino acid carboxyl group loses one proton to form carboxylate ion, and NH2 amino group accepts one proton to form NH3 positive ion. So a dipolar structure is formed called zwitter ion or internal salt structure. So this zwitter ion, in this zwitter ion form, amino acids reacts with both acids and bases because it acts as amphoteric, or you can say it shows amphoteric behavior in its aqueous solution. So let's see first in acidic solution. I have already written the formula of amino acid, and if you react with acid, then in acidic solution it will take proton, and this carboxylate ion will accept this proton from the acid and will form a structure like this. This will remain same, and here a proton will be accepted by carboxylate ion to form COOH, and the entire structure now formed is a positive ion, or you can say a basic ion. So in acidic solution, amino acid exists as a positive ion or basic ion and forms salt with acids. Similarly, in the basic solution contains hydroxyl ion, and hydroxyl ion reacts with the proton loosed by NH3 positive and form NH2 group here. A proton will be released from here and will combine with OH negative to form water, and now here you will get H2N, CHR and COO negative. So here a negative ion is formed because H proton is lost from this end, and it will form a negative ion, and this negative ion or acidic ion is formed in the basic solution and form salts with alkalis. So in acidic solution it exists as a positive ion, and in basic solution as a negative ion. Now next term is isoelectronic point. If you pass electric current through this solution, you can see this in acidic solution positive ion is formed, so it will migrate towards cathode, and in anode in basic solution because an anion is formed in the basic solution. But at a particular pH at which the amino acid should not migrate to any of the electrodes and exists as a neutral dipolar ion is called isoelectronic point. In acidic solution it will migrate to cathode, in basic solution it will migrate to anode, but at a particular pH at which it will not migrate to any of the electrodes, that point is called isoelectronic point, and it depends on other functional groups present in the amino acids. Like in neutral amino acids, the pH is usually from 5.5 to 6.3. Okay, and at isoelectric point, the amino acids, they are very less soluble in water, and therefore it will help you to separate different amino acids in a polypeptide chain or in a protein, क्योंकि protein में बहुत सारे amino acids होते हैं. So it will help in the separation of different amino acids of a polypeptide chain. Next is accept glycine. All the naturally occurring D amino acids are optically active because of the presence of this chiral carbon atom or asymmetric carbon atom, and therefore it exists in D and L form that I have already discussed in the previous video also. If amino group is on right side, then it is a D form, and if it is on left hand side it will exist in L form, and all the mostly naturally occurring amino acids, they have L configuration.